What's up, everyone? All right, so we're back here. So it's been quite a long time actually since the last time I was doing this experiment. So full disclosure, it's been well over a month since these samples have been actually growing inside of these petri dishes. And a big reason why I've taken such a long time to make this video is it seems like the bacteria inside this petri dish has taken a very long time to grow. And I suspect that it wasn't, I suspect that it wasn't because the genes aren't that dirty or because they didn't have bacteria. I suspect maybe that during the process of me collecting the samples that, maybe I made some mistakes in terms of how I was collecting the sample, or maybe there was something that I wasn't doing with these petri dishes. So it's taken well over a month in order for me to get any type of substantial results. So let's go ahead and take a look. As you can see here, again, we started out with the control. These were brand new genes that we tested to see if they had any bacteria on them. The second one was the actual dirty genes themselves. And then after that, what we did was we put the genes inside of the freezer for longer than 24 hours. And then we took a sample. We let those genes then warm up to a room temperature, and we took a sample. And then last but not least, what we did after that was we then steamed the genes and then took a sample. So after waiting over a month for the bacteria to grow inside of the petri dish, what are the results? And let's go ahead and take a look. So these are the control genes. Let's go ahead and flip this over. Now down at the very bottom there, as you can see, we don't have any type of colonies growing, so there's no mold in that one. So that one was pretty much expected. We'll put that off here. The dirty genes, as you can probably see from the very top there, we got a little bit of something there. Let's go ahead and flip it over. So as you can see here, we definitely have some growth inside of this petri dish, which, as you would assume, there was definitely something that we were going to pick up when we took a sample for some dirty genes that I haven't washed in months. So yeah, and one thing too, obviously you can't get this from the video, but there is the smell of mold, and you can actually smell it from all of these petri dishes. You can definitely smell mold. So so after this is over, I'm definitely throwing these out. But as you can see here, we got some mold growth. We'll go ahead and put that off to the side. Now let's take a look at the cold genes. Now these are genes that were put in the freezer for longer than 24 hours. And let's go ahead and take a look at the results. And as you can see, we do still have some bacterial growth inside of the petri dish here. And let's compare the last one. Remember this one here, the dirty warm genes, room temperature. And then these are the ones that were put in the freezer. Now, as you can see, there's slightly less mold growth or bacterial growth in there. So freezing your genes does help a little bit, but as you can see, you still have some bacteria that's on the genes there. Next up, these were the sample was taken again after the genes had warmed up after being in the freezer for 24 hours. And let's take a look. And as you can see there, we still have some mold growth. So even after being in the freezer for longer than 24 hours, as you can see, there's still mold growth. And let's actually compare the two. So again, the one on the left here was placed in the freezer for 24 hours. We then took a sample immediately after we're taking it out of the freezer. This is what we got. And then after it being left out for half an hour, or I should say it was left out so that it was room temperature. And we got another colony here, which is a little bit surprising, but I'm sure biologists will tell you that some bacteria will actually go dormant when they're in a very cold environment, and then once they warm up, they can actually start to reproduce again. So it's not really surprising that the genes that were placed in the freezer for longer than 24 hours still had bacteria on it, so that one was to be expected. And last but not least, as you can see, we have these steam genes. So on this one, we took a pair of genes, steamed them, Now we're trying to see what the results are, and then this one is really surprising. So if you turn it over, as you can see, there are no colonies. There's no bacterial growth anywhere in this petri dish. And what we'll do is we'll then take a comparison to the warm genes. A little bit of bacterial growth. The cold genes. The dirty genes. And our control genes. So the ones that are definitely the biggest difference, these ones are the cold, or the genes when they're the dirtiest at the room temperature, and then after being steamed. So if you're somebody like myself who goes many wears in between washing your jeans, the quickest way, easiest way of being able to preserve the dark wash in your jeans is to just steam your jeans. So after I did this experiment, I actually steamed my own jeans, and it really just kind of gives you that feeling back, like it's a refreshed pair of jeans, and it's something I highly recommend. And then, as you can see here, the results speak for themselves. That actually steaming your jeans is a lot better than placing them in the freezer for longer than 24 hours. So again, the results speak for themselves. You're kind of wondering what's the best way to get all the germs off of your jeans in between wears, is just to simply steam them. I know it's kind of an interesting experiment. Many of you out there are probably thinking it's pretty messed up that people can go months in between wearing their jeans, or I should say washing your jeans. But the best way of really kind of making sure that you're somewhat sanitary is to just to steam your jeans.